Thank you to the organizers for the chance to present work today that was jointly supervised by Marinka Zitnik at Harvard Medical School, James McFarland at the Broad Institute, and William Hahn at Dana-Farber. The key idea of the talk is that words and genes share a correspondence. Their meanings arise from their context. For example, apple is a polysemic word whose particular meaning is resolved via sentence context. Similarly, H2AFX is a pleiotropic gene whose particular function is resolved via cell context. So depending on the cancer drug used to treat a cell, H2AFX will activate different DNA damage repair pathways. While unsupervised learning of word polysemy is common, unsupervised learning of gene pleiotropy is unsolved. So undergraduate ML courses will teach that a corpus of sentence contexts can be used to learn word embeddings that obey linear semantics. Is there an analogy in genetics? So the goal of today's talk is to address unsupervised learning of gene pleiotropy. The data that we propose using for this is gene perturbation effect measurements. The way that these effect measurements are gathered is by treating a starting cell line with CRISPR-Cas9 guide RNAs, and then observing the phenotype over time. So for instance, if a guide RNA knocks out an essential gene, that guide RNA will deplete from the population, resulting in a negative score in this sort of fitness effect here. And it's now very popular to repeat this over many cell contexts. So for example, many cancer cell lines or the same cell line treated with different chemical insults. And we think this is a good starting point because this is a causal effect measurement of the action of that gene on cell physiology. There are other alternatives that we considered, but this is probably the most direct measurement. For the approach, we're interested in low-dimensional vector embeddings. 2011, Wagner and Zhang published an influential review of pleiotropy in which they highlighted 3 criteria for thinking about pleiotropy in high-dimensional phenotypic spaces. The first is that gene representations ought to be sparse. Biologically, genes tend to have a few functions, not too many, and not 0. They also recommend that the latent variables that you learn from the high dimensions align to biologically meaningful constructs like protein complexes or pathways. And finally, they recommend accounting for redundancy between cell contexts. Motivated by these criteria, we developed a method that we call Webster, which learns a dictionary matrix that sparsely approximates gene effects. So the setup is that we take as input this numerical matrix. Rows are cell contexts, columns are genes. Each entry is the essentiality of that gene in that context. And from that input, we learn sort of a low rank matrix here that we call the dictionary matrix, where now every column, instead of being a gene, is now some kind of biological function learned from the correlations between genes, essentially. And for each function, we have an inferred fitness profile over those same biological contexts. The second matrix is this loadings matrix. So the task is for each input gene effect, learn a representation of that gene effect as a mixture of up to T of the elements in the dictionary. And so T is a parameter that you can choose. And this is the overall reconstruction task or approximation task that Webster is assigned. So we added a few augmentations to help preserve interpretability. As I alluded to just a moment ago, there's correlations between gene effects that are highly informative of their biological functions. And this has been widely studied now by many groups. And so we can be aware of this graph of correlations between columns in the original matrix as we learn our representations. So we want to encourage, let's say, 2 genes that are highly correlated in their input data here to have very similar loadings onto these functions that we learn. We can do a similar trick to help manage redundancies between cell contexts. So 2 cell lines or cell contexts can be highly similar in their gene effects. If that's the case, we want to preserve that similarity when we learn this low dimensional representation of this fitness data set in terms of functions. Putting it all together, Webster is a method that takes as input this fitness data does a lot of pre-processing, standardization, eliminating genes with no phenotype. And that pre-processed matrix serves as the input for this task. The outputs are this dictionary matrix here and this loadings matrix here with the key hyperparameters controlling the size of the dictionary and the degree of sparsity in the loadings matrix. The objective function is as follows. So the first one is very standard for these tasks, just trying to approximate the original data in terms of these low rank matrices. Additionally, we augment this objective with a graph prior over the columns of X that is induced from the correlations between columns in Y. And similarly, we impose a graph prior on the rows of D based on the correlation structure between rows in Y. We applied this method to real world data. This group here, the Durocher lab, published a group of screens that they performed in the same cell line under different genotoxic conditions, such as UV light. And these treatments cause DNA damage that need to be repaired in order to keep dividing. So by doing a genome scale screen in those treatments, they were able to uncover which genes are necessary for survival in the presence of some treatment. So that purple here, if this negative fitness effect, means that gene is critical for surviving whatever DNA damage that treatment induces. So Webster takes as input that matrix here. So we filtered it to the top 300 high variance fitness genes, and there are 30 treatments here. And taking only the numerical data as input, Webster reconstructs that data in terms of a dictionary of size 10, and in each gene effect approximated as a mixture of 2 dictionary elements. And these hyperparameters were chosen via grid search. You can see that in our reconstruction, we capture the broad patterns present in the data. So one of the outputs, as I mentioned, is this dictionary matrix. It has 10 columns. Each column contains the inferred fitness effect of knocking out some latent entity. Here's our loadings matrix. As promised, it's very sparse. Each column only has 2 non-zero entries. Now this becomes most interesting when you start to align it to gene sets that were hidden during model training. So these authors nominated literature-validated nucleotide excision repair genes. And you can see that in an unsupervised way, this first latent variable that we learned seems to map to a cluster of these kinds of genes. And so that motivates our naming of this function over here as nucleotide excision repair. If we look at the dictionary element over here to which that corresponds, UV seems to generate a negative fitness effect in the absence of nucleotide excision repair. That makes sense because UV is makes DNA adducts that are excised. So how does this model, how does this model handle pleiotropy? Well, let's give an example. So H2AFX is a DNA damage response gene that has a fitness effect when you knock it out in certain treatments. So that measurement was approximated as a mixture of 2 of the dictionary elements in our screen, learned from the screen. The first is a dictionary element that captures the effect of homologous recombination. And the second is a dictionary element that captures the effect of end joining. And these are the specific treatments that cause a fitness effect on those pathways. Now, if you recall from the introduction, this is the pleiotropic example I started with. So H2AFX encodes an ensemble of DNA damage functions depending on the input. And of course, this was hidden during model training. Finally, we can do some of these linear semantics. So this H2AFX measured fitness profile, which is measured over 30 or so treatments. If you subtract our dictionary element of representing end joining, add a dictionary element corresponding to Fanconi anemia, you actually get a vector size 30 or so that is very similar to RAD51B. And this makes sense because RAD51B is a mixture of Fanconi anemia as well as homologous recombination. So here's that synthetic vector I just described, plotted against the measured RAD51B fitness effect over each of those treatments in their screen. So Webster also scales to larger data sets. This is a data set called dependency map in which 675 cancer cell lines were screened genome-wide. And so by taking this matrix and decomposing it, we get a dictionary matrix of size 220 and a loadings matrix with 3,000 genes. These are the high variance genes from this data set. And T being set to 4, meaning that each gene effect is approximated as a mixture of up to 4 functions. And again, these were chosen via grid search. So once again, genes obey linear semantics in this data too. So SHOC2 is a signaling protein downstream of upstream signaling factors. And in the model, its measured fitness effect over cancer cell lines was approximated as a mixture of 4 dictionary elements that were automatically learned from the data, each of which captures the effect of one of those pathways. So to look at global relationships sort of at a bird's eye view, we can make visualizations of this data by taking each gene effect and embedding it in some UMAP space or 2D plot. And similarly, because we learned these functions and each function has an inferred fitness effect over these contexts, we can jointly embed them into that same plot. So here's the result of doing that. Here are 3,000 high variance fitness genes that were the input to Webster. You can see that they're non-uniformly distributed in this embedding space. And now when you overlay those learned functions, you can see that they act as cluster centroids. And this is exactly what's happening in high dimensions. Gene effects that are collinear will usually collapse down to a single latent variable that we call a function that's automatically learned by the model. These are highly interpretable factors or functions, and so you can kind of explore this. Here's an example here where we learned 6 different independent biological functions capturing aspects of the mitochondria, but overall they sort of group over here with their constituent genes nearby. So once you learn this dictionary, you can perform a few tricks with it. For example, if you've done a compound screen over cancer cell lines where you've measured the sensitivity of each cell line to that compound, you can actually take that profile and project it linearly into this latent space that we've learned automatically from CRISPR screens. And what that will do is it will basically align your compound to the closest genetic function that it thinks your compound might be involved in. And that usually helps with recovering this mechanism of action. But I'm going to conclude today's talk by talking about my favorite application, which is biological discovery. And if you think about why physiological co-relationships occur, oftentimes it has something to do with the physicality of these 2 genes. And oftentimes you can recover protein complex relationships this way. So I'll give you an example of how pleiotropy helps with thinking about protein complexes. So switch-SNF is a protein complex that's mutated in 20% of human cancers, and it's highly modular. So there's an enzymatic module that binds to 3 different versions of the complex that have different functions on the genome. In our model, out of the 200 or so dictionary elements, 3 of them actually map to these 3 different versions of switch-SNF. So if you look at the gene effect models, ARID1A, for instance, only binds CBAF. So in its loading structure, that's reflected, where it has a strong loading on the CBAF factor or function, but 0 for the others. On the other hand, SMARCA4 binds all 3. And if you knock out SMARCA4, you incur a fitness cost that's a mixture of all 3 of these complexes, which is exactly what we get in our model. This is also true for the integrator complex. When we first started looking at these Webster factorizations, we were surprised to find that 3 different ones seem to map to integrator components. When the structure came out last year, we actually noticed that indeed it was picking up on structural modules that we just didn't know about because the structure wasn't published. In particular, this INS10, 13, 14 heterotrimer is of note. So C7orf26 is a gene of unknown function. And if you think about its loading structure, 3 out of its 4 loadings seem to map to some version of the integrator complex, with a particularly strong loading on this INS10, 13, 14. And so we had a hypothesis that this might be a specific part of that module. And indeed, when we went to the lab and I did some biochemistry to figure this out, C7ORF26 binds and stabilizes INS10, which allows it to bind the 13-14 heterodimer to form what we now think is an integrator heterotetramer module. And so in this specific case, the loading structure was reflective of the underlying stoichiometry. So to conclude, unsupervised learning of gene pleiotropy is tractable. The data I recommend using are these high-dimensional gene perturbation readouts. And if you're interested in these linear semantics, consider using sparse approximation. So I'd like to once again acknowledge my mentorship team and open it up for any questions. So thank you for your time.